So a lot of you are not in your best mood, and I completely understand that. So, but I just posted. I'm not sure that whether you have seen or not, but in the social platform, I just put a note, a couple of lines in there that, as I just kept saying on the first class, is it's not really easy to give 100%, and you sometimes have the heartbreak. And this is one of the things that I just wanted to say, that if you just, I don't know whether you have seen that, just after the game, did you see the Twitter from Dabo? The thing is that he acknowledged that they were better, they played on the other hand. And did you see the coach's reaction in Ohio State in the last one? He was angry, he was mad. So that makes a huge difference, that how you approach, and that says a lot. That's why he's a winner. So with that, let's. So just a couple of things before recording. I do have a voice throat issue, so I will do this a while. I'm sorry for that. I have an itchy throat. A couple of things that I'm just trying to still play, that what is the best way to do this. And what you're going to see, that I have uploaded the video in your Canvas, and you can see that there are two parts, part 1 and then part 2A. Pretty much, if you just combine the lecture time, the video time of that, that's pretty much one lecture. So that's how we will do that. And then I will upload, preferably, if not tonight, definitely by tomorrow morning, by before noon, the second part, the Thursday's material. So I will upload that. And what you are going to see, that I constantly make changes to the PowerPoint slide. And what you're going to see, that I initially tried to put the slide like part 1, part 2. I think that that's going to become a really, like you have 10, 20 files, and it's really messy. So what I will do, that I will just put just one file and name it as a experimental technique in cell biology slide set. So now you have the set up to today's material, totally you have that. And then I will add up the material on behind that. So you will have just only one file, and you just download a save as, and it will just replace that file. And what you're going to see, that sometimes while recording my lecture, I see that there are not enough information in there, and I just modify the slide. And you will see that in the slide there is a note that on this, like a star marks, which say the slide denotes material added change from the lecture videos. So I will explain those in the class as well. So the first chapter, sorry for the delay, what we're going to talk about is the culturing and the visualizing the cell. And along with that, we will also cover the part of the light microscopy, which will be the bright field microscopy, and that will be pretty much the first material. So as you know, that it is already a flipped class, so I will not spend that much of time on the very basic material. It will be very repetitive. So just overview of that the scientists, when they work on the cells, they first need to actually grow them in culture. And there are two ways you can do it. So the first one is actually the primary culture, where you can actually take the cells from a tissue and then you actually take them apart or separate them out into a heterogeneous population of cell. And from there you can plate them and grow them. Now this will be a mixed population. And we call this one, as you know, that is a primary cell culture, which will need some of these material. So here just a couple of things that I have added here, and if you're just curious that what I have added is just the last part. So you know that you need the proper condition, rich media. I'm not going to read the slide because it's very self-explanatory. Antibiotic and the antimycotic, the reason that we need to constantly, and this is an issue, that why we use antibiotic, antimycotic, because even if the complete sterile environment, you're working in a hood and completely clean environment, but still there are airborne bacteria, fungal spores floating around, and all it needs is just one bacteria or fungal spores going into the flasks where you're culturing, and they will take over, and you cannot, they will not survive. So because the animal cells probably will grow like doubling time is about 20, 24 hours, whereas some of the bacteria will grow in 20 minutes. So they will take over, and that's the reason you use that. So one of the important things is that many of these animal cell culture actually do need a platform, which is pretty much the bottom of your culture vessel, whether it's a round dish or flask. And these cell adhesion molecules, which are the CAM molecules, these include the collagen, glycoproteins like fibronectin, laminin, glycosamine glycans like hyaluronic acid. These are some of the attachment molecules. So this is here, this is important information, and these could be your recap type of questions for your quizzes and exams. So here we are looking at the primary culture that we are talking about, that you take the tissue and from there you take the culture, and they often differentiated properties of the organs from they were isolated. And you have already seen this video that these are the myoblasts, which are basically the precursor stem cells of the muscle cells. And from there, they can differentiate into long muscles. You can see that link in your video as well. So I'm not going into the detail. On the other hand, this is the example of that. So on the other hand, fibroblasts secrete collagen. Nerve cell extend axons that are electrically excitable as the synapses. You can easily see those. So those are very general idea. So here there are just two definitions what we mean by that cell strain. When we actually mean a cell strain, that you have a primary culture collected from the tissues, and from there you can pass them a certain amount of time. And as I said, I already used the word pass them. What we mean by that pass or the passage, that means that the subculturing or splitting of the cells from one flask to the other. Or any one culture vessel to the other culture vessel. And you can see that video that how it's been done, that you need a couple of different things, that the cells are going to attach themselves on the bottom of the flask and they're not going to come out due to the CAM, cellular adhesion molecules, the CAM molecules. So you need to usually use a chelator, like a calcium chelator, like EDTA. And along with that, you need to use some protease or proteinase. And usually the most commonly used are, for this purpose is trypsin. And along with that, you can also use collagenase or combination of both. And then the cells will, it is very clear in the video, so look at the video at your own time, that you will be able to see that the cells will actually come out from there. And then what you do that from one flask, you actually make a dilution of the cells and you pass it to the next one. And there is a formula, you do not have to remember the numbers, that how many cells that you put per square centimeter, usually it's 2,500 to 5,000 per square centimeter. That's the number of cells that you put in a new dish. And within 3 or 4 days, they are going to become confluent. We are using a word confluent, means that they are going to cover like a lawn the bottom of that flask or the dish, and then you got to passage it over and over. So you constantly have to do that. So one of the things that when you actually, if you work in a lab, you're going to see that all these medias are actually, most of the cell culture medias are red in color. Can anybody tell why it is red? Yeah. What? Yeah, that is correct. Absolutely. There is an indicator which is phenol red, which will turn into an orangish color, orangish yellow color when the pH actually drops. So that's pretty much, you know that when you actually start, it is red at neutral pH. Then gradually as you grow the cell, it's going to become towards the yellowish, and by the 3 day when the cells are confluent, it's going to drop the pH of the media and you have to change them. So it's a visual indicator. Very good. All right. So after that, we are going into that. The primary cell actually do have a finite growth cycle, and it sometimes could be 50. At the most, it is 100 generations. So this actually gives you a kind of an interesting idea that in your body, a single cell actually don't divide more than 50 or 100 times. That's it. After that, that cell is done. So that actually keep check any problem or damage to the DNA as well, because the more generation it passes, the chances of mutation in the DNA increases and it can become a cancerous cell. So this is actually an autonomic way that the cells are actually programmed to die after certain divisions. So essentially when you actually start a culture, You start with a rapid growth rate, it will rapidly grow, and then it will kind of level off. And 50, sometimes could be 100 generations, the cell are going to die, and we call this an senescence. So that is the limitation of the primary cell culture. The primary cell culture cannot grow for indefinite time. So if you have a particular tissue that you do not have any other kind, you have to rely on them that at a certain point of time, you have to have the new tissue and extract the cells from there. So these are some of the limitations. On the other hand, this is another type of cell, which is the transformed cells, and these are extremely interesting. So what happens that how you actually get the transformed cells. So let's go back to the very first one, and if you just read a book about Henrietta Locklear, have you read the book by yeah, so it's a very interesting story that Henrietta Locklear was a patient at the Johns Hopkins in the 1950s, and she went there with a cervical tumor, and the scientists and the physicians actually took her sample of the cells and put it in the culture, and it started grow indefinitely. And by far, you can just easily Google out that HeLa, H-E, then capital L-A from Henrietta Locklear from that patient. That is the most used cell line in cell culture. There are a couple of more. And what is the beauty about it? Those cells were actually isolated in 1951. This is 2020. Thousands of laboratories across the world use that cell. They don't stop growing. And this is what is called a transformed cell. It is an oncogenic transformation. There was something that happened into the DNA that will not allow the cell to stop growing. And essentially, there are some of the proto-oncogenes activated, or tumor suppressor genes silenced. These are some of the ways that you actually get these transformed cells. And in the laboratory, the scientists actually develop these transformed cells. So usually what they do, that they will use a chemical mutagen, which is normally it's a carcinogen. It's harmful. But they will add it to the media thinking that there could be some mutation by which they can actually transform into this immortal cell line that will never die. And sometimes some of the viruses actually do that. Some of the retroviruses actually do this oncogenic transformation as well. So in those cells, you will initially, what will happen, that there will be an initial loss of growth potential, and it will go into a senescence right here. And then from suddenly, if you're lucky, you're just looking at your cells every day, and most of the time you are just probably getting frustrated that, okay, all right, these cells are all dying, that's nothing happened. And then just one day you have a magic bullet that one cell suddenly started to emerge as an important cell line, then you have already obtained a cell line as such. So this is actually the most commonly used in the laboratory. Primary cell cultures are not used. These are actually used in the laboratory much more often as well. And these are some of the characteristics, and these are very self-explanatory, not going into that. And a couple of examples are there. HeLa, I just put it on the first. Then HEK293, Chinese hamster ovary CHO cells. So all these are mammalian cells. This is from the kidney. This HEK is a kidney cell. On the other hand, there is just only one cell that I put. There are many other. This SF9 is an insect cell. And it is actually isolated from an insect called Spodoptera frugiperta. It's a fall armyworm. It's a caterpillar. So from there, this is actually isolated, and this is a beautiful cell, large cell compared to a mammalian cell, grows. And this is actually used for growing many of these recombinant proteins. And the beauty of this SF9 cell is that they are not attachment dependent. All the insect cells can grow in culture. So you can take a large vessel like a fermentation flask type vessel. We call them as a spinner flask, and you can aerate that and grow these cultures as well. Those are some of the characteristics that some of these cells which are transformed actually don't depend on attachment as well. So this is a drastic improvement over the regular primary cell culture, because you can, if the growth is not attachment dependent, you can grow them in a suspension culture, means you can grow them in a larger density. And if you are expressing a protein or recombinant protein through them, they will actually produce way more compared to, and also will reduce the cost of the flask and the dishes. So these are some of the consideration as well. So the last part of this type of cell that we are going to talk about is this embryonic stem cells. And then just remember that there is just one word that we actually use. These are fastidious. That means they require fastidious in terms of that they require specific growth factors and material to grow. Otherwise, they're going to die. It's much more difficult to culture them. And usually what we do that you actually get them from this ICM, the inner cell mass from a blastocyst. And that's one of the reason that people think that this is an embryo. And when you kill them, that's kind of an ethical issue arises from there. That's a controversy for the stem cell research as such. Now, these embryonic stem cells, you have to use some feeder cells because they are going to provide the hormones or the growth factors in there. Now, if you remember that I did not touch that, if we just go back to this slide, there is something that we talked about. See, that in the rich media, we have essential amino acid vitamin, but after that, you have a peptide and protein growth factor, often supplied by the addition of the serum, which is a fetal calf serum. So we need the growth factors, but that is very defined. You can have the bovine serum to supply them for the growth of that. So you need that. That is actually the most expensive component of the cell culture. A 500 ml will cost you in between $500, like 500 ml, $500, or even more sometimes. So very expensive. That is the most expensive part of the culture. So here, on the other hand, you need the similar type of organism cells like a feeder cells of fibroblast. Sometimes if you just want to have a cleaner physiology, that you are just actually looking at these embryonic stem cells, thinking about that the physiological condition that you are measuring is not exclusively from the embryonic stem cells, but you are measuring the activities or the metabolites from the fibroblast. Then what you do that you have a clean flask or culture vessels where you have only the embryonic stem cells, and along with that you just add these factors, leukemia inhibitory factors, WNT, bone morphogenic factors. These are all transcription factors, and these will turn on some of the specific genes that is needed for the growth of these cells. So that is, remember that they contain the DNA for everything. But those needed to be turned on or express the proteins, what is needed for them to support their growth. So very fastidious in that process. Now what we are going to do, the first question, clicker questions, let's do that. So we have about 211 people in there already attended. So let's do the first one. Transformed cells, such as cells derived from the tumors, have a finite lifespan. We'll go with easy. I mean, like, all the questions today are really, I mean, like, is this all about? I mean, like, you are not going to expect these questions in your exam, though. It's going to be a little bit difficult compared to that. I think we are good, about 205 responses. Anyone else? Just waiting about a minute. All right, a minute. Let's take a look. All right, so after that, what we are going to see, that now you have this group of cells. Now, we are going to isolate them. Why we need to, that suppose you have started your culture with a primary cell culture, which is not a cell line, and then we are going to isolate them because this is a mixture of a cell. When you actually isolate them from a tissue, it's very difficult to get a homogeneous population of tissues. So how you can do that, it's very easy. Think about that when you are drawn the blood, you have red blood cells, white blood cells, different types of white blood cells, and the doctor actually tell the pathogenist or writes a prescription that you actually do have to run something which we call as the differential count. So the differential count, what it means by the differential count, that you have to use a centrifugation technique to separate your red blood cells and the white blood cells, because they have different densities and they will actually precipitate at different centrifugation speed. That's how you can actually separate them. Physiological property, affinity for glass macrophages. This is actually one of them. And then the last part is the flow cytometry. And the material that is added, which is not in your slide, if you're just looking at the slide now, is that what you can actually achieve from the flow cytometry. So these are some of the things that is good for your quiz questions, that you can actually measure the cell shape and size as well. But you cannot check cell density. This could be a quiz question. You cannot check cell density as such, that how compact it is or how loose it is. You cannot check that, but you can check the shape and size. You can add a dye which will directly bind to the DNA, and the DNA will fluoresce, so you can measure the content of DNA in the cell. And what you do, that depending upon the cell surface, you have all the different antigens, and if you have a specific antibody to specific cells, you can actually separate them. And the beauty of this system is that, think about that, nowadays we are also going to describe a very simple machine for that, but there are some of the sophisticated machine that what we call as a 7-channel or 10-channel FACS or fluorescence activated cell sorter. What we mean by that, simultaneously it can actually project or receive 10 different signals at 10 different wavelengths. So you can have a mixture of 10 different cells. It is so sophisticated. 10 different cells, 10 different antibodies. Each one of them is conjugated with a different dye. And you have 10 bins, and you can isolate all of them 10 at the same time. These are $1 million, close to $1 million instrument as such, but you can do this with their FACS. So this is some of the basic idea, that here you can see that there is a T cell. If suppose you are just isolated the spleen and crushed it up, and you just want to isolate these T cells from there. So to do that, there is a process where you have conjugated these two antibodies on which are on the surface of the T cells. So the T cells actually have this Thi2, which is the thymocyte 1.2. That's actually an antigen. And you have a CD3. These are the thymocyte 1.2 and the CD3. You actually raise antibodies against that we'll talk about somewhere else. So you have these specific two antibodies, and you have a red fluorescence conjugated to the antibodies. This is a chemical reaction, and a green fluorescent dye conjugated to this protein. So you can just label it just singly. You do not have to label them doubly. You can just label them singly. And on the other hand, these are the other immune cells which are not labeled. Now you actually homogenize the spleen from after this, and then load it onto a flow cytometer. So here, what you are seeing in the flow cytometer, you have a cell suspension fluid, which is mixed with a sheath fluid, which actually don't mix. The cells are in the middle, and the nozzle through which the cell actually pass is close to the diameter of one cell. So only one cell actually pass at a time, and you actually shine a laser beam onto that. And what happens, there are several different types of detectors available. One is a scattered light detector. So this will actually tell you the size and the shape of the cell, and if there is a cell there or not. So that scattered light detector will do. On the other hand, when you supply the laser beam, it's going to actually fluoresce those dyes which are attached with the antibody, and that will be directed by this fluorescence light detector. So again, we will talk about the fluorescence, that how it actually works. Usually you just have a typical wavelength given to the dye, and it's going to emit a different wavelength as such, which is going to be detected. Now what happens, that right here, there are some plates and very sophisticated instrument. Depending upon the fluorescence that is being detected by the machine, it's going to add a small amount of charge onto surface of each cells. And the machine is so sophisticated, depending upon the intensity of the fluorescence, it can vary that charge as such. And this is, we are talking about in the millivolt range. One is charged at, say, 10 millivolt, another is charged at 12 millivolt. and the machine will be able to distinguish these two. So it is extremely sophisticated in that process. Then what's going to happen, that if there is a charge, then the cells are going to deflect in another way. If there is no charge, that means there is no fluorescence as such, which are the blue cells, which are the splint cells, doesn't have any fluorescence. So the machine did not put any charge onto that. They will all go into the bin, which is discarded. On the other hand, if the fluorescent molecules actually is having the charge, they are all being collected. And if there are different types of molecules with different fluorescence in terms of intensity or color, they can be collected on different beams because they will be deflected by another plate, and that small charge difference can differentiate them or put them in a different place as such. Very sophisticated instrument, and again, just the principle of is important. I'm not going to talk about the physics or the instrumentation or the engineering of that as such. So this is actually an important slide that definitely you will encounter this type of question in your exam for sure, critical thinking question, that how you read a fax or a flow cytometry data. So what you're going to do, that here we are just looking at just a 2 die based data. Sometimes you can have another axis, but 2 die based data means like right here you have the origin. As you go along the x-axis, that means the intensity of the red fluorescence is increasing along the x-axis. And as you are going through the y-axis, the green fluorescence, which is conjugated to the Thi1,2, is actually increasing. So increasing green fluorescence, increasing red fluorescence. Everybody good with that? So now let's go back to this slide and see that what happened. The blue cells don't have anything added to it, so it is not going to fluoresce. It's not going to fluoresce red. It's not going to fluoresce green. On the other hand, the T cells are attached with a red dye as well as the green dye. They are going to fluoresce red and green. So what you are seeing right here, you have the cells which have very low fluorescence, almost none. These are your non-T cells or the splint cells, which are not tagged with anything. On the other hand, if you just go, this means red fluorescence higher. If you go, red fluorescence higher. If you just go right here, green fluorescence higher, that's it. So the T cells actually, as they are conjugated with red and green, they are found in this quadrant. On the other hand, now the question could be, this is your derivative question or application-based question, that there could be a mixture of 3-4 different type of cell. Suppose each of them is so suppose you have a cell here which is only conjugated with a green fluorescence, you will find it here. If it is only conjugated with a red fluorescence, you will find it here. If they are both tagged with the both fluorescence, you will find them here. If you don't have any conjugated dye to that, you will find them here. So this is basically how you read a simple fax data. And the quadrants are actually just conventional, that how you label them. It's quadrant 1, 2, 3, and 4. I actually had to take the quiz question away because I was just using some publisher's question. And there was a question on this where they say that where you're going to find these cells A, B, C, and D, and none of the quadrant was labeled. And I'm like, okay, all right. So there are already a couple of different questions, and we're going to address those in there. We have some issues in there. So I'll open it up. Don't worry about it. Just making some corrections. All right. So that's about your fax and the flow cytometry, how it is done. Now let's take a look at these growing cells in a 2D or 3D culture. So far, what we have seen, that you have a flask or dish, and all the cells are growing on its surface. But see that this is a new image that I have added. That's why you have that blue star in there. If you are culturing epithelial cells, they have a distinct surface. You can see that right here you have an apical surface, which is the top. The main function for these is a secretion absorption. Think about the cells of the lining of your intestine. This is the apical surface which is facing the lumen or the tube of your intestine. And this side, the basolateral domain, is actually the attachment surface. So it is very difficult for the cells, if you actually allow them to grow on a single plate or something like that, they actually don't differentiate properly. So here what you have, that you have this 2D culture system where you have a porous base. So you have the media coming from the bottom towards the basolateral domain and the top. And why it is convoluted? Because that's basically showing that those are the microvilli, which increase the surface area, which is common in many of these cells. That's why they are showing these. That's the apical surface. And the epithelial cells grown by this way will behave more normally because it's going to mimic the condition in the tissues, at least to some extent. On the other hand, this is also, again, even much more advanced, that the 3D printers are now using this pliable matrix, which is extremely porous, and you put those blocks and the stuff in the culture flask to grow these material. So here you can see that you have a soluble gradient is absent, whereas here you have a soluble gradient. And the cells here in the 2D culture, you just have a top and bottom, but you really do not have a 3 dimension. Here the cells can polarize in 3 dimension. What do you mean by polarization? So a cell actually, your apical is one pole, your basal or lateral is another 2 poles. So when they can differentiate in characteristic that there is a character of the apical one or basal one, that means the cell has a polarity. And what you are seeing in this picture, these are made in Darby canine kidney cells from a dog, a fully polarization potential for that. So what you are seeing right here, that these are actually the cells which actually line the kidney tubule. So if you take a kidney tubule, the kidney tubule, you take a cross section. So right here in the middle where the pointer is, this is actually the hollow hole of the, so if you have a pipe-like structure, this is basically the lumen of the pipe, and this is the lumen of the tube. So the cells with this red fluorescence are basically facing this lumen. That's the apical surface. So these cells are stained by a fluorescent dye specific to an apical surface protein. On the other hand, this green is basically the lateral and the basal surface specific proteins as such. And the blue thing is the dye. It's a DAPI, which is a nuclear dye, showing the nucleus. So you can easily see that when you are growing these cells in a 3-dimensional culture, you can almost mimic a condition which will give you a tissue-like representation. And a lot of people are actually working on these and even culturing organoids or similar culture. So here is another thing. It's fascinating if you just look at that. People are actually, suppose somebody actually lost an ear. I'm just saying because I just saw this, ear in an accident or something like that. So the scientists, they are printing a ear with the 3D matrix, which is this pliable matrix. They're putting that in the culture so that it grows the cell on that, and they can put it back to the patient. The technology is going to that efficient. I mean, like, it's not a science fiction. You can look it up in the internet, that how they're trying to do this artificial organs by this process. So this is actually one of the most important part, and we'll spend some time in this part, that the hybridomas actually produce monoclonal antibodies. And why we are actually, why we are emphasizing this while we are talking about the cell culture, because this is one of the prized thing that we actually can take advantage in the cell culture. And these monoclonal antibodies are of tremendous importance, not only the biological sciences research, but particularly cancer therapy. If you actually have a particular mutation and have a protein expressed, you can target drugs for that particular protein expressed on the tumor cell using this monoclonal antibody. So what it is. So suppose you have an antigen which is X. This antigen is a protein. Now the most important part, and this concept, just make sure that you understand this, that a single protein, which is an antigen, have a 3-dimensional surface, which will have lots of epitopes. So just think about that if this is a protein, you have some epitopes on this side, some on the top, some on this side. Each one of these could be an epitope. This could be an epitope. Epitope is nothing but a stretch of amino acid, which is giving a typical 3-dimensional structure on the surface of the protein, not inside. On the surface of the protein, which can be actually recognized by either an antibody or a B cell receptor to which it will receive an antibody. So one single protein will have multiple epitopes. A monoclonal antibody is basically if you are raising an antibody against a single epitope. So this is a whole protein. If you inject this whole protein, suppose antigen X, you will have antibodies in the mixture in your blood of this epitope on the side, top. You will have a mixture of these antibodies. These are called the polyclonal antibodies, tremendously important because think about that. If you have a virus, does it have multiple antigens and you want different antibodies to bind different places so that it can be neutralized faster? Huge therapeutic purpose. But what happens that out of those, if you have selected just only one epitope like this epitope, and if you can target an antibody against that, it will only recognize to that epitope. Think about that. In a cancer cell, a protein mutated, and this is the only one which is different from the normal protein. You can target that specifically. It will not touch any other cell. The other cells have the other epitopes, not that. The cancer cells have only that epitope. So you can target that specifically, and that's the beauty of the monoclonal antibody. So how you do this? So essentially, for this purpose, you actually inject the mouse with this antigen X, the whole protein, you do that, and the mouse is going to raise antibodies, polyclonal antibodies, of all mixture. Here we are just only considering the red epitope, that we just one. But it will be a mixture of many other, and the blue cells, suppose those are the splenic cells. And now you take those cells and you are going to fuse them with these myeloma cells, and this is a cell line. So this is an immortal cell line, and you are going to fuse them. Usually what you use polyethylene glycol. This is a fusing agent, or Sendai virus. They actually do have a protein which actually, cytomegalovirus even, they can fuse the cells. So now you have a hybrid cells, and eventually their DNA, their nucleus is going to merge together as well. So now you have a fusion, and now after fusing the cell, we actually transfer them to a media which you call as a HAT media. This is a selective media. This is a little bit of an addition to what you have in your textbook. You don't have that in there. This is called a hypoxanthine aminopterin thymidine media, HAT media. So this is a selective media, and it's going to select the cells with the specific B cell producing that antibody and the cell which is fused to that, so it is immortal. All other cells are going to die. And now what you can do, that when you are seeing that there is a small B cells on a dish or a flask, you can just isolate just one cell. Suppose you just put them in a new flask where they are all separated. You can just put a clonal cylinder, which is a hollow glass tube, open at both ends, and you just put that on the dish. So some cells, B cells, are going to go inside that. You take out those cells. That is from one clone of B cells. Now you propagate that. That's an immortal cell line. Will produce a monoclonal antibody for years, indefinitely. So how this selection works? So here, the selection, how it works. So first, what you have, that the B cells, all B cells that you're going to isolate, they will actually have a specific enzyme, which is thymidine kinase, or again, another enzyme can be used instead of that, which is the hypoxanthine guanyl phosphoribosyl transferase. So any of these two can be used. So in the HAT media, any of those. So the right here, it will have the TK as well as this AGPRT, which is not shown but is written here. This will have both of them in their DNA. So what happens that, on the other hand, here, the immortal HAT-sensitive plasmacytoma, these are the myeloma cell line, they are thymidine kinase negative, they cannot express that, and AGPRT negative as well, right here. So these cells, although they are immortal, as they cannot use these specific two material, either hypoxanthine or the thymidine, these are the two intermediates from where you actually make your nucleotides. So this is a nucleoside as such, and this is also another one. So from here, you will just make your nucleotides. So in this media, this cell will not be able to make the nucleotides. This cell will die. On the other hand, this cell will be able to utilize those to make the nucleotides. But see that the B cells have a mortal lifespan because they are primary cells. They are not going to live forever, so they are going to die as well. On the other hand, what you're going to see that the fused cells will survive. So you have the immortality from the plasmacytoma and the deficiency of either the thymidine kinase or the HGPRT is rescued by the DNA which is coming in here. So these are the only cell which are going to survive. So these are your myeloma cells that's going to grow over as such. And now you can just select just only one cell from that culture and you can culture that. That's your monoclonal antibody production. Tremendously important. Hybridoma cells grow rapidly, are immortal, produce the monoclonal antibody encoded by its B lymphocyte, are formed by fusion of a normal antibody producing cells, all of the above. Already 1:15, so let me stop this one. So let's take a look at this. So E is the correct answer. All of the above are actually true. So that's good. All right, so the next question is the monoclonal antibodies bind one epitope on an antigen, can be secreted by cultured cells called hybridomas, are important for basic cell biology research, can be used as therapeutic agents, all of the above. That's it. So you can see that these are really easy questions. We're just still running this in a trial mode, so just bear with me. So let's take a look at some of these ideas. This is just a very general idea that many biological processes can be studied in the cell culture as such. And here you can see that there are selected drugs that can be used in cell biology research. So the thing is that am I going to ask you to memorize all of them as such? Probably that will be kind of like really it's not, but just remember just a couple of important ones here. Briffeldin A inhibits Golgi function, and it's hugely important when you try to use the secretory function of a cell. Cytochalasin B inhibits actin, and that's going to destroy the cellular morphology because it's a cytoskeleton. And inhibits the microtubule, the nocodazole. These are some of the important ones as such. And another important one that I just want you to just probably, that if you're just working in a lab, that may actually help to some extent. If you're just looking at one of the protease inhibitor, if you look at that right here, there is something which is called phenyl methyl sulfonyl fluoride. Phenyl methyl sulfonyl fluoride, or PMSF, that is very commonly known. So if you are working in any kind of lab or something, this is probably the one you are going to use, and that's the only one that I will just put that one in here when I upload the slide, but that is the only one probably you just need to from here. And I'm just not going to ask you too much questions from here other than the important ones which is mentioned here. So here is a classic example that how you can actually use the cell biology study to actually scan through the material and it actually isolate a drug which will do some specific things. So this monoastral is a particular drug which actually disrupts the microtubule. That means that the cell will not be able to divide. And this is a classic example of that process that we have seen. So with that, we will just end the part one and we will just go into the microscopy. And the thing is that why I kept so much less time, because this you are studying from your high school. So we will just talk about a little bit of the specific material as such. Not going to go into the detail of that. Development of light microscope. And here there are a couple of things that you need to remember, that the objective lens actually magnifies your image 100 times, whereas it would be again magnified by your ocular, or we call it projection lens, sometimes called ocular even called eyepiece, and eyepiece, and you just multiply these two numbers, so that this is the total will be 1000x. So this is how you actually calculate that. So the next one, which is important, that this could be a little bit different, that the resolution is again another important factor for these microscopes. And you cannot actually see any object which is lesser than, there is a formula that your wavelength of the incident light that you are using is very important for the resolution. And let's take a look at how this actually works. So for example, for the visible light, about 0.45, so this is wrong. I'm sorry, I'm going to change that. So 0.45 micrometer is, this is not millimeter, this is micrometer, I will change that. 0.45 micrometer is the wavelength of violet. 0.7 micrometer is the wavelength of the deep red. So if we are using the microscope in this visual range, see the resolution is, on the other hand, is dependent upon this. And we are going to talk about that, how this limits the light microscopy set by this wavelength. So let's take a look. So the resolution, again, one, is the minimum distance between the two distinguishable objects. If it is 0.5 micrometer, and you cannot resolve the objects which are apart than 5 micrometer. So how this actually is calculated, so this is the formula, and you will have application type of questions in your exam on this one. So your D, which is the resolution, is 0.61, which is a constant of lambda. This is the wavelength, and over n sine alpha. So what is alpha? Lambda is the wavelength that you know that will be given to you, say 0.45 micron for violet light. So this is actually the objective of the lens, and this is the stage. Now, right here, this is the angular aperture, and this is actually the half of the angular aperture is the alpha, which is right here. So the total angle is 2 alpha, half is alpha. So how this alpha is determined, this is actually by the manufacturer of the microscope. Everybody is trying to push this limit to go over 70 degrees is the best. I mean, like, if somebody can make it 72 or 74, that's an extremely great microscope. I mean, like, common microscopes can have 50, 60 angular aperture. The more the angular aperture, more expensive is the microscope as such. So they try to push this limit as such. And then what you have, that the N right here is the refractive index, which could be the air, which could be water, which could be anything like immersion oil as well. So when you actually plug into this, you can see that we are looking at the shortest distance, 0.45 micrometer, best media immersion oil about 1.5, and maximum alpha is 70 degrees. So if you look at that, you can actually resolve it to 0.194 micrometer, which is about 2 micrometers. So that's actually very specific as such to push this limit. So you can see that that is about 0.2 micron as such. So that is the limitation. So think about that. No microscope can give you anything more than 0.2 micron apart. The rest will be digital magnification. Your optical magnification, no matter whether it's a $20 Toys R Us microscope or a $200,000 microscope, your optical resolution is limited to 0.2 micrometer. Now the computation have pushed that limit. We're going to talk about that later. And I know that we are just going a little bit slower. We just have to adjust that time in the first 1 or 2 classes. But let's do the last clicker question. And with light microscopy, if the objective lens closest to the specimen magnifies 40-fold and the eyepiece lens magnifies 10-fold, then the final magnification will be 440, 400, 4000. So that is correct. You can easily see. You can see that the question that we just asked, that is the correct questions. 400-fold is the correct answer. So bright field microscopy is the first one of the optical microscope, and you can see that these are some macrophages, and you can see, I mean, like, it's not the very impressive image that you can actually see. So these are some of the limitations. Advantages is that simple, inexpensive, no staining required, but there are some limitations. And we're going to push these limitations and see that how we can improve the regular microscopy. So the first thing that what you have, this is an important thing, that you can use a phase contrast microscope. So what it does, and these are the only two things you have to remember from here, that here you have an annular diaphragm right here. Whereas on the top, you have a phase plate. So these are the two buzzwords in here. You have an annular diaphragm and a phase plate. So what it does, that due to this, when the light passes through the specimen, depending upon the refractive index, it splits them into two different phases. What we mean by the phase, suppose this is, I'm just trying to say that this is the crest of a wave, and rather than aligning the next wave with the same refractive index in a similar way, it is pushing it a little bit apart. So the light waves are coming to your eye or the projection lens not at the same phase, but a little bit different phase. The wave is actually shifted about 1/4. And that is good enough. I mean, like, not going into the physics, but that is good enough to actually produce some of the images like this with a phase contrast microscopy, which is a huge improvement of the bright field microscopy to the phase contrast microscopy. Then what we are looking at, that there is another type, which is this differential interference, or DIC, also known as Nomarski interference. So how this actually works, it works on a polarized light. Think about that you can easily see that the polarized light actually takes out the oscillation of the waves from different directions. Think about that. You have a Ray-Ban polarized sunglass. You just put it on, and you just immediately see the glare is gone. So a lot of bouncing light bouncing around, and your polarization actually takes those bouncing light. So these all beams are coherent, going in the parallel direction in one direction. So this is what the polarizer is doing. And then it passes through a Wollaston or Nomarski prism right here. And this prism is below the condenser. This is below the condenser, which actually puts the light beam onto the image. So what happened, this Nomarski prism actually split the light beam in two orthogonal complexes. So one light beam going in there, it splits it into half. And each one of them is going to be split in half. So it is just, this is just only one beam, but every one of them is going to go in half. So when they are going to pass through the specimen slide, think about that you have the specimen somewhere on that slide. So these waves are going to, two different waves are going to go through the specimen with different refractive index, and some of them are going to go around the specimen just through the glass slide. So what's going to happen, that you actually are going to see a difference in these phases. So how you actually ultimately see that? Above the objective, you have the second objective, which is a Nomarski prism right here. This actually converges these two orthogonal complexes back into the same waveform. So you initially actually split them, then you actually put the wave back together. This is called the constructive and the destructive and the constructive interference. And this is going to give you a contrast, which is going to give you almost a three-dimensional image of the material. Very different looking as such. So what you're going to see right here, you can see that in this picture, the DIC Nomarski, you almost have a shadow against a background. And this, particularly, this is great for looking at the shape, overall shape of the different cells. So this is a classic example of DIC Nomarski interference. Now, all these microscopes actually doesn't have to be different. You can just have one bright field microscope, and you can just put this ring right here, which you have like right here. This is the bright field opening for your condenser. You don't need anything. Right here, these are all your Nomarski DIC or the Nomarski prisms right here. All of these are. And these are your annular rings. You can see right here, there is an annular ring. This is smaller, this is larger. Depending upon the objective size, you are going to change that. So the question would be why you have 3 prisms. You have one for 4x, one for 10x, one for 40x. Depending upon the aperture of the lens, this actually varies. So you have all your DIC, your phase, all of them all together. So this is the beauty of this system. So the next clicker question, and the last one of the day, is going to be, you have a bright field light microscope and want to retool it to make a phase contrast. What parts do you need to buy? Annular diaphragm, condenser, filter, light source, phase plate. You can have more than one answer. This not allowing you to? Uh-oh. So I have to put this as a quiz instead of a poll, and then I can choose to. I'm not going to go into that. I think that's good enough. All right. So actually, that is a correct answer. I mean, like, if you just, can't I do this? So can you see that A and E is both chosen in your thing, but you couldn't answer A and E both. You couldn't, right? Okay, I will just have to move those in there. So that is correct. So A is correct. You need an annular diaphragm, and this annular diaphragm is at the bottom below the condenser, and you need a phase plate as well at the top. So these are the two. So A and E is correct. So don't worry about that if it's marking you wrong. Both of these are actually correct answers. All right. So let's take a look at this again. Annular diaphragm and the phase plate. You can see that these are the two. So all these questions will be in your this I will upload again, so you will be able to see that. So here we are looking at some of the sample preparation as such. So the fixation is that you actually going to keep these cells frozen in time. You use glutaraldehyde or formaldehyde first to fix the cells so the components actually don't move. There are some limitations to those, but you have to fix the cells before you visualize them. And then if the cells are not in culture, which you can visualize directly onto a slide, you have to put them in an embedding media. A very common embedding media is paraffin wax. You can just put it in there. Or sometimes for electron microscopy, you use epoxy resins, which are a chemical reaction. You put it in there and it will polymerize. So many different types of media are available. Once you have that, then you put them in a microtome. The machine is called a microtome. Sometimes it's called a cryomicrotome, where the sample, if it is a paraffin or a wax block, is actually frozen so that the slices are very, very correct. This is like a daily slicer. So you just slice them, put them in a thin slice, and put them on a glass slide, and you process them, and finally you stain them. So the cell staining, so first what you are looking at, the hematoxylin, and you have to stain them to have a contrast. So hematoxylin is a basic positively charged dye, cationic stain, and usually that will actually color the nucleus. Eosin is an acidic, negatively charged ionic stain which binds to these basic amino acid like lysine, arginine. So your pink will be, your cytoplasm will appear as pink. So this H and E is actually a very common stain routinely used by all the pathologists throughout. So very common. This is not in your book. This is a Wright Giemsa stain, histologic stain for blood and bone marrow cells. The reason I put that, because you are going to see this Wright Giemsa stain in many biological samples. You're seeing a sample in your anatomy and physiology and any other classes that what are the different stains. You can tell that this is a Wright Giemsa stain. That's why I put it in here. So this is a mixture of these two different type of dye, and methylene blue and eosin. And the nuclei stain blue-violet, cytoplasm is a pale pink, erythrocyte plain pink. So this is one of the classic example of that. On the other hand, see that this is also another very powerful thing, benzidine, which binds to the heme-containing protein. So this is actually a Xenopus embryo, which actually do have an island. This is a blood island. What you mean by the ventral blood island, all the blood cells are accumulated there, which contain the heme for the hemoglobin, and that actually specifically stains them. So that's pretty much, so we are within time, 1:41, so we did pretty all right with the timing. Now, one thing before you wrap up with that, whether it's your online book or even your print if you have a copy, the material that we covered today, there is a drastic mistake in a few lines in your book. You need to find that and post in your course discussion. It's a very major mistake. We have discussed that here, and you are going to find the information that you are going to get in the book is different from what we put. Let's see whether you can find it or not.